In this video, we're going to be looking at some details of a couple of different processes, one of them being replication of DNA. That means the copying of the DNA that occurs in the nucleus. The second process is actually where DNA is starting to do its job in the cell, and it's a similar process to replication, so we're going to talk about them at the same time. These notes are going to go on page 9 in your notebook. Remember the structure of DNA. We've been talking about it for a while. Remember the two strands of DNA are held together by hydrogen bonds, adenine bonding with thymine and cytosine bonding to guanine. Now, it's really important to remember that those are hydrogen bonds. We talked earlier in the year about hydrogen bonds and how they're not very strong and they're easy to break, and this is going to be important in what we're talking about. Here's just a reminder of what the structure of DNA looks like. Here's sort of a simplified version where you have your bases in the center, and then if you look up close, you can see more detail. You don't have to know all of this, but you should notice that here, running up the middle, are some hydrogen bonds. This little red dotted line represents hydrogen bonds holding together guanine and cytosine, adenine and thymine. Now remember, whenever the cell is getting ready to divide, during the S phase of mitosis, it's going to be copying the DNA. And so we're going to talk about the actual process of copying the DNA in this lesson. We call it DNA replication. And because of the structure of DNA, it's real easy because, of course, the cell can just kind of peel that DNA open at the hydrogen bonds. And now you've got an open piece of DNA that can be copied. Now, DNA, they say, is a replication of DNA is semi-conservative, which simply means that the old strand is a template for the new strand. So whenever the DNA is copied or replicated, you end up with two new DNA molecules, each consisting of half new DNA and half old DNA. So it looks something like this. If we start out with this DNA molecule, half red, half blue, you kind of open the thing up, and that serves as a template or like a guide for how to copy it. You end up with two new strands. Half of this strand, the blue, was from this original strand, and then the other half is this orange new one. Same thing down here. You have the original red from the original DNA, and now you also have this new blue one. So that's what we call semi-conservative replication. So let's talk about now how it works. You start with some enzymes called helicase. Helicase is an enzyme. Its job is to open up the DNA. So if this is our DNA ladder, helicase enzyme comes in and peels this thing apart. We say it's unzipping because it kind of looks like a zipper. But basically what it's doing is breaking apart the hydrogen bonds between the bases in the center of the molecule. This forms something called a replication fork. That's what's located right here in the center. Once you have the replication fork, then another enzyme comes in, DNA polymerase enzyme, polymerase, and it's going to bind to each strand of the DNA and begins building a complementary strand. So wherever there's an A thymine comes in, a T comes in. Whenever there's a C, a guanine comes in. If there's a G there, a cytosine comes in. Okay, so the base pairs tell the molecule kind of how to come in. The DNA polymerase is the one that brings them in and makes them bind together. The new strand, again, like I said, is built according to base pairing rules, adenine with thymine, guanine with cytosine. On one side, the leading strand, they call it, the building is continuous. So every base just keeps coming in and base pairing, and it just gets all matched up. On the other side, it's called the lagging strand. It's only copied bit by bit in these little fragments. And there's a reason for that. It's because of the way that the DNA has to be copied in one direction only. So in the lagging side, because it's in pieces, there's another enzyme, DNA ligase, which comes in and it stitches them together. So it's in these little pieces, and then DNA ligase puts in the missing pieces to stitch it all together to one nice strand of DNA. So it looks something like this. Here we have the helicase that's unzipped the DNA. We have the leading strand where the bases are just coming in and making a nice strand here with DNA polymerase. In the lagging strand, it's making little fragments because it has to be copied in the other direction, so it has to copy just pieces at a time. These are called Okazaki fragments, which is kind of a cool name, but that's what they're called, these little pieces. Eventually it gets pieced together by DNA ligase. So now we're going to switch gears. That was all about DNA replication, how DNA is copied. Now let's talk about what DNA is doing. Its job in the cell is to contain the code to make protein. So we keep saying DNA is the molecule of life and it's so important, and Watson and Crick won the Nobel Prize and all of that. But what is it really doing? Its job, it contains the code to build protein. Remember, protein is a sequence of amino acids. Each stretch of DNA, then, we call that a gene, and each gene codes for a particular protein. It contains that code. But because DNA has to be protected in the nucleus, in a eukaryotic cell, of course we're talking about eukaryotic cells, the DNA has to stay in the nucleus. So the code from the DNA for a particular gene, in order for a protein to be made out there in the cytoplasm, the code has to be somehow copied and then carried out and then translated into protein. So from nucleic acid, it has to be translated into amino acid. Okay, so this is kind of an overview of the process. We start with DNA, and then we have a process called transcription, which I'm going to talk about in just a minute. And we make something called RNA. And then we have another process called translation, which is literally translating nucleic acid in RNA into protein. So let's talk about this process, transcription. Transcription is making a copy of the DNA to carry out of the nucleus. It's copied onto not DNA, but it's copied onto another nucleic acid, so it's a similar structure. It's called RNA. So instead of deoxyribonucleic acid, it's just ribonucleic acid. Very similar, but different in three ways. RNA, instead of being a double strand, it's a single strand. So it's like half a DNA molecule. And instead of thymine, RNA has this other nucleic acid, this other base called URACIL. So wherever you have adenine, instead of bonding with thymine, it's bonding with URACIL. So you'll see some U's instead of T's. And then RNA has ribose sugar instead of deoxyribose sugar. But for the most part, you still got your phosphate, you still got your sequence of bases, you still have base pairing just like you did with DNA, but RNA is just one strand. So if we were to look at it, here is our DNA molecule up here, and it's got two strands with hydrogen bonds in between, and here's our code, T with A, C with G, G with C. RNA here, I'll tell you what that M's for in just a second, but RNA here is just like a single strand, like half a DNA, and you have your sequence, again, of bases. Notice there's U's there instead of T's. URACIL instead of thymine. Now, the process of transcription is going to look just like replication that we talked about a minute ago. We have helicase that unwinds the DNA, it unzips it and opens it up. Then we have, instead of DNA polymerase, we have RNA polymerase. RNA polymerase comes in and begins to build the RNA molecule, just like with the DNA, according to base pairing rules, cytosine with guanine, and instead of adenine with thymine, we now have, of course, uracil. When finished, the RNA is going to break away from the DNA. It's going to leave the nucleus, and it's going to go into the cytoplasm. Okay, so that's the whole process. The DNA unwinds. The RNA is made with RNA polymerase according to base pairing rules, and then when it's finished, the RNA is going to break off and go out of the nucleus into the cytoplasm. This finished RNA is known as messenger RNA, or we call it mRNA. And we call it that because it's carrying the message out of the nucleus. So finally, here's sort of what the process might look like. You have your DNA molecule. It's being opened up here, and you've got the code. RNA polymerase enzyme is going to read the code and start to build this messenger RNA, okay? This messenger RNA, this red strand, is going to leave the nucleus and go into the cytoplasm, and then the next lesson I'll tell you what happens to the message from there. That's it for this one.